Hi guys, I hope you'll benefit from the review classes during the prep week. Since most of the students find it challenging to get their heads around the anatomy of the nervous system, especially the differences between the somatic and the visceral innervation and how they interconnect with each other while innervating various parts of the body, and also considering the student feedback as well, I thought I might as well take on the task of describing the nervous system anatomy to you guys, and then extend this knowledge to discuss in detail the somatic and the visceral innervation for the respiratory, GI, and the cardiovascular. Right, so as part of this presentation, I'll review the structural and the functional divisions of the nervous system. Then I'll briefly allude to the meningeal wrappings of the brain and the spinal cord, like the pia, arachnoid, and the dura mater. Next, as part of the peripheral nervous system, I'll discuss the spinal nerves and the cranial nerves, their formation, their exit points from the CNS, and their functions briefly. Next, as part of the functional division of the nervous system, I'll elaborate upon the functional anatomy of the somatic and the autonomic nervous systems. We'll see the structural and functional differences between the sympathetic and parasympathetic nervous systems as well, and understand the route the autonomic neurons adopt as they go on to innervate various body parts. Well, you all know by now that the nervous system comprises of a central nervous system and a peripheral nervous system. The CNS, the central nervous system, comprises of the brain and the spinal cord. These are the control centers of our body, and being the vital organs, these are protected inside the respective bony cages. The brain inside the cranial cavity and the spinal cord inside the vertebral canal. Now let's have a look at this cartoonic illustration over here. See, this is it's a mid-sagittal section taken through the head and neck area, then through the vertebral column. And what you can see over here is the brain residing inside the cranial cavity, and this is the mid-sagittal section of the vertebral column. And you can see the bodies of the vertebrae in the front, so that's the ventral aspect, and these are the spinous processes of the vertebrae. And the spinal cord can be seen nicely resting inside the vertebral canal over here. The CNS is then connected with the rest of the body via nerves, the spinal nerves and the cranial nerves. These form the peripheral nervous system, the PNS. Once again, let's resort to this cartoonic illustration over here. Here we're looking at the base of the brain, and you can see the brain stem continuing down as the spinal cord. So you're basically looking at the ventral surface of the spinal cord over here. So you can see that there are nerves coming off from the spinal cord. There are total number of 31 pairs of spinal nerves which come off from their respective spinal cord segments. So that means there are 31 spinal cord segments. These spinal nerves, they then exit through the intervertebral foraminas and then innervate different peripheral structures. And similarly, we've got 12 pairs of cranial nerves which more or less pop off from the base of the brain. These then exit the cranial nerve, the foraminas located inside the cranial cavity, and then become extracranial, where they innervate the head and neck structures, the thoracic and the abdominal regions. Right, so let's talk a little bit about the connective tissue wrappings of the brain, which are known as the meninges. Now, the brain, as well as the spinal cord, they're both they're covered up by three connective tissue wrappings, which are called the meninges. The outermost connective tissue wrapping, the outermost meningeal layer, is known as the dura mater. So what you can see over here is basically the superolateral surface of the brain, this being the frontal aspect. This would be the posterior or the occipital lobe of the brain. And you can see that there is a thick white tough membrane covering over the brain, and that's why you cannot see the sulci and the gyri underneath this thick tough fibrous membrane. This is known as the dura mater. And you can see the same dura mater over here as well. Here you're looking at the medial surface of the brain, and this being the frontal aspect, and this would be the posterior or the occipital lobe of the brain over here. And you can see on the medial side as well, there is dura mater covering over the brain. Once again, you can see the sulci and the gyri underneath. And then if we remove the dura mater, then here you can see that the dura mater has been removed, and now you can see a relatively more fine, delicate, transparent membrane covering over the lateral surface of the brain, and this is basically our arachnoid matter. And you can see once again, you can see the arachnoid is jumping from one gyrus onto the other gyrus, so it doesn't really go into the sulci and just wrapping over the brain completely. And this is kind of one common feature which is between one feature which is common between the dura mater and the arachnoid matter. And the common feature is that they both basically, leaving aside a few exceptions, they jump from one gyrus to the other, don't go into the sulci. And you can see the same arachnoid matter over here as well. See, the dura mater has been removed, and now you can see the arachnoid matter covering over the cerebral cortex underneath the dura mater. If I remove the arachnoid as well, which obviously I can't show you over here, but if you remove the arachnoid membrane, then underneath that we'll see the third meningeal covering, which is the vascular pia mater. This is the vascular layer, and it actually really tightly approximates the gyri and the sulci. So that's the only layer which is going to be seen, which can potentially be seen inside the sulci. So the three connective tissue wrappings of the brain and the spinal cord, those are the dura mater, followed by the arachnoid mater, and then the pia mater, which is the innermost layer. So this is a slide which shows you some prosections of the human brain. The brain is the part of the CNS which lies within the cranial cavity. The outermost surface of the brain is called the cerebral cortex. This is the most advanced part of our brain, which differentiates us from the lower mammals from an evolutionary perspective. Think of the cerebral cortex as a large sheet of gray matter, and if you need to fit this entire large sheet of gray matter into the tight confines of the cranial cavity, then naturally it has to be folded, and because of this folding we see different elevations and different depressions on the surface of cerebral cortex. For instance, we can see an elevation over here, you can see an elevation over here. These are known as the gyri, and the sulci between two successive elevations, these depressions are known as the sulci. We can see a sulcus here, we can see a pretty big sulcus over here. The two major sulci are the central sulcus and the lateral sulcus. Central sulcus can be seen here, color coded in red, and it separates the frontal lobe in the front from the parietal lobe of the brain at the back. The frontal lobe is concerned mainly with the motor functions, parietal is concerned mainly with the sensory functions. Then we can see a lateral sulcus over here, which is color coded in blue, and you can see it separates the frontal lobe and the parietal lobe at the top from the temporal lobe of the brain down below. Lastly, we've got the occipital lobe or the occipital cortex at the back, which is concerned mainly with the vision. Then here we can see a mid-sagittal section through the brain, through the cortex, and we can see the inner parts of the brain over here. We don't need to go into the details. We'll talk about that in detail in third year. What we can see is the brain stem coming down from the brain, from the cortex, and we can see that the brain stem has three different subparts to it. The midbrain here, the bumpy part over here, which is the pons, and then that continues down as the medulla oblongata. That is, the medulla oblongata is the area where the vital cardiorespiratory centers of the body are present. Behind the brainstem, we can see the cerebellum, which is concerned with motor coordination, balance regulation. It is in fact the posterior relation of the pons and the medulla. The medulla then continues down as the spinal cord. Right, so now that we know that the brainstem continues down as the spinal cord, let's talk in a bit more detail about the spinal cord. So starting with this prosection, the picture of a prosection over here, what we can see here is that, see, the part of the cranial vault has been opened up. You can see that the meninges have been removed as well, and you're basically looking at the brain from the back over here. So these are the occipital lobes which are concerned with the vision. Beneath the occipital lobes, the cerebellum has been removed, and you can see the back of the brainstem, because we know by now that the brainstem is the anterior relation of the cerebellum. So if I remove the cerebellum, I can see the back of the brainstem. And then I can see the brainstem, the medulla oblongata here, passing through the cranial cavity and continuing down into the vertebral canal as the spinal cord. So we can see that spinal cord nicely resting in the vertebral canal over here. Have a look at this prosection here. Now this is actually a mid-sagittal section through the vertebral column. You can see the bodies of the vertebrae over here and the intervertebral discs in between the successive vertebral bodies. And so this would be the anterior aspect here. These are the cut sections of the spinous processes of the vertebrae. So therefore, this would be our vertebral canal, and you can see the spinal cord nicely resting inside the vertebral canal. Now, if you look at the entire the length of the spinal cord, you'll it's a bit hard to appreciate over here, but the diameter of the spinal cord is not really constant throughout its whole length. There's a bit of an enlargement in the cervical region, which is called cervical enlargement, and an enlargement down below in the lumbosacral region called the lumbosacral enlargement. And the reason why the spinal cord is a little bit enlarged in these regions is because there are extra neuronal cell bodies and neurons in these regions which are going to go to the upper limb from the cervical enlargement and to the lower limb from the lumbosacral enlargement. So there are more neurons in the spinal cord, the cell bodies in the spinal cord in those regions, and consequently a bit large sized spinal cord can be seen. The diameter is a little bit enlarged in these regions, therefore. Right, so now have a look at this illustration here. Now this is once again another cartoonic illustration of a mid-sagittal section through the vertebral column, and we can see the bodies of the vertebrae in the front. So this would be the anterior side. You can see the spinous processes and then the back of the sacrum over here. What you can see is the spinal cord is ending roughly at the L1 vertebral level in adults, and this cone-shaped ending of the spinal cord, this region is known as the conus medullaris. It's really important to know this region from a clinical perspective as well, because there are fecal urinary control centers in this region. But anyways, this cone-shaped ending is the conus medullaris. And in case of kids, the spinal cord can extend a bit further down below as well. But what you can see is that there's a bit of an empty space over here, or at least the spinal cord is not down below in the vertebral canal over here. And even though the spinal cord ends at this level, the meninges still go down all the way here. And so this area would be filled in by the CSF, the subarachnoid fluid, and the cerebrospinal fluid. And we can actually safely do a lumbar puncture over here without risking any damage or injury to the spinal cord, which is going to be higher up. Then what you can see is that there are spinal nerves coming off from the spinal cord segments, and even though the spinal cord ends at the L1 level, the nerves still have to go down before they can exit through the respective intervertebral foramina. So consequently, these hanging bunched up nerves, these are known as the cauda equina, and that is the reason why the nerves kind of bunched up over here, because the spinal cord is ending higher up. The nerves still have to travel a longer distance. Now, these things can be nicely seen over here as well. This is actually a prosection of the spinal cord. You can see the meninges have been removed so that we can see the spinal cord nicely. This is the cone-shaped ending of the spinal cord called conus medullaris. And you can see all these spinal nerves coming off from the spinal cord and kind of hanging down over here, bunching together to form what is known as the cauda equina. Right, so let's shift gears a little bit and let's talk about the peripheral nervous system. The peripheral nervous system comprises of the cranial nerves and the spinal nerves, and we can talk a little bit about the cranial nerves first. So there are 12 pairs of cranial nerves, and you can probably see in this cartoonic illustration over here that all of those cranial nerves, they are originating more or less from the base of the brain. The prosection over here also shows the base of the brain, and you can see several cranial nerves popping off from the base over here. And we're going to use this cartoonic illustration and prosection interchangeably to identify those nerves. One good thing about these cranial nerves is that they pop off from the base of the brain in a sequence. We start from the front, from cranial nerve number 1, and if we make our way backwards, we're going to end up somewhere on the brainstem with the cranial nerve number 12 over here. Right, so if we look at the front over here, this is more or less the undersurface of the frontal lobe, and we can see the olfactory nerve over here, which is the first cranial nerve that is concerned with the sense of smell. We can see in this prosection the olfactory nerve is shown over here, and this is the olfactory nerve on the other side. So these are pairs of cranial nerves. There's going to be one cranial nerve on either side, just like we've got pairs of spinal nerves. Then if we make our way backwards over here, we can see the optic nerve over here. So whenever on an illustration or in a prosection you find this X-like structure over here, this is basically the optic nerve, and there are details associated with it, but I'm just going to simplify it for now. Let's just call this as optic nerve, although different parts of the X have different names to them. This is the second cranial nerve, and this is concerned with the sense of vision. Right, so now let's make our way backwards and downwards, and now you can see in this prosection, you can see this is the brainstem, and the first part of the brainstem, which we can see here, is the midbrain. It's not really clearly visible in the prosection because it's all covered over by loads of little vessels, but you can see the midbrain very clearly in the cartoonic illustration over here, and you can see that there are two cranial nerves coming off from at the level of the midbrain. This one over here from the front of the midbrain is the oculomotor nerve. That is the third cranial nerve. Remember, we're moving in a sequence, so this is the third cranial nerve, and then this is the fourth cranial nerve called the trochlear. Trochlear, or the fourth cranial nerve, is the only nerve which originates from the back of the brain stem. Everything else comes from the front of the brain. We can see the midbrain over here, and this is the level where the third and the fourth cranial nerves are going to come out from. They're not clearly visible on the prosection though over here. Then if you make our way downwards over here, this is the pons, right? And you're looking at the ventral surface of the pons, and this is our fifth cranial nerve, or the trigeminal nerve, which comes off from the front of the pons, and we can see that trigeminal nerve over here very clearly, and this one over here. This is a mixed nerve, and it takes sensory information from the face and is responsible for innervating the muscles of mastication, so it helps basically with the chewing process. So that is our trigeminal nerve, or the fifth cranial nerve. The cranial nerve number 3 and 4, the oculomotor and the trochlear, which we discussed, those are both concerned with primarily with moving the eyeball. So they innervate the extraocular muscles responsible for the movement of the eyeball. Then if you go further down over here, this is the area which is basically demarcating the junction of the pons at the top and the brain stem and the medulla oblongata down below. And you can see that there are 3 cranial nerves coming off from the base of the brainstem in a sequence moving from medial towards lateral. These would be cranial nerve number 6, 7, and then the 8. Cranial nerve number 6 is the abducent nerve. The cranial nerve number 3, which is the oculomotor, and the trochlear, which is the fourth cranial nerve, and the sixth nerve, which is the abducent, they all innervate the extraocular muscles, so they kind of share a common action, which is the movement of the eyeball. Then if you go more laterally from the sixth cranial nerve, this is the seventh nerve, which is the facial nerve, and one of its main functions is going to be innervating the muscles of facial expression. And then lateral to it is our eighth cranial nerve, which is the vestibulocochlear nerve, and that has 2 main functions. It is concerned with the sense of hearing and is concerned with the sense of balance. So it is responsible for carrying the neuronal information concerned with the sense of smell and with the balance regulation. We can see the pons here on the prosection and the medulla oblongata here. So this is basically the junction of the pons and the medulla, and that's where the cranial nerve number 6, 7, and 8 are going to pop off. We can actually see the 8th cranial nerve over here on the prosection. Cranial nerve number 6 and 7 are not that clearly shown here. They're probably broken by the time the brain the prosection was taken out of the cranial cavity, perhaps. So then if you go further down over here, we can see cranial nerve number 9 and 10. 9 is the glossopharyngeal nerve, and 10th cranial nerve is the vagus nerve. They both innervate more or less the areas in the larynx and the pharyngeal regions. And remember, we're moving in a sequence, so after 8 comes 9, which is glossopharyngeal, and then 10, which is the vagus nerve. Then if you go further downwards over here, we can see the little rootlets of the 11th cranial nerves coming off from over here. This is our accessory nerve, and that is concerned with innervating two muscles, the trapezius and sternocleidomastoid. And if you go a little bit to the front over here, then these are the rootlets of the 12th cranial nerve, which is the hypoglossal nerve. And the hypoglossal nerve is innervating primarily the muscles of the tongue. If we look over here, this is a cartooning illustration once again of the base of the brain. So now we've actually kind of reviewed the basic features. We've identified the 12 pairs of cranial nerves. We have briefly talked about their functions. So how about if we just pause the slide over here and just review the cranial nerves one by one at your own pace and try to quiz yourself and see if you actually remember the names of the 12 cranial nerves and their functions. Right, so now that we have reviewed the cranial nerves, let's talk a little bit about the spinal nerves. So what you can see over here is a horizontal section of one of the vertebrae. We can see the vertebral body in the front. The spinous process can be seen at the back. This is the vertebral canal, and we can see the spinal cord resting inside it. If you look at the horizontal section of one of the spinal cord segments, you can see a butterfly-shaped gray matter. Gray matter is nothing but the accumulation of neuronal cell bodies. The anterior horn of this butterfly-shaped gray matter is motor. The posterior horn can be seen over here, which is sensory. The spinal nerve is formed by the union of the anterior and the posterior roots. The anterior root is the motor root, which means it contains the motor neurons. The motor neurons, they travel through the anterior horn of the spinal cord, travel through the anterior or the motor root to reach the spinal nerve. The posterior root is sensory, which means that the sensory neurons coming from the spinal nerve, they're going to pass through the sensory root or the posterior root and then bring that sensory information into the posterior horn of the spinal cord. We can also see a dorsal root ganglion over here, which is nothing but the accumulation of neuronal cell bodies of those sensory neurons bringing sensory information into the posterior sensory horn. Now what we can see here is that the anterior root is purely motor, right? The posterior root is purely sensory. However, when the two roots combine together to form the spinal nerve, then the spinal nerve is a mixed structure. And when I say it's a mixed structure, then it basically means that it contains both sensory as well as motor neurons. The spinal nerve then exits through the intervertebral foramen and splits up into rami. So we have an anterior larger ramus and we have a posterior ramus which is smaller in size. The anterior ramus innervates the anterolateral body wall and maybe the limb musculature and the tissues of the limbs, the upper limbs and the lower limbs, and the posterior ramus innervates the posterior body wall. Unlike the roots, the nerves and the rami, they are all mixed structures. So they contain, just like we discussed, they contain both sensory as well as motor neurons. Now let's take an example. Let's say a sensory information of pain has to come in from the back and go to the spinal cord, the central nervous system. That sensory information would be carried by a neuron, a sensory neuron running inside the posterior ramus, bringing that sensory information from the receptors on the back. Those sensory neurons are going to travel through the spinal nerve, then go through the dorsal root ganglion, through the posterior sensory root, and bring in that information into the posterior sensory horn. Let's say for motor information, the motor command has to be sent from the central nervous system, from the spinal cord to muscles of the upper limb. Then a motor neuron will pop off from the anterior horn, travel through the motor root, and then through the spinal nerve and travel through the anterior ramus, through the plexuses, brachial plexus, and they're going to go and then innervate. Those motor neurons are then going to go and innervate the muscles of the upper limb. Now let's have a look at this 3D cartoonic illustration of the spinal cord. Once again, you can see the gray matter over here. We can see the anterior horn, which is the motor horn, the posterior horn, which is the sensory horn. We can see the sensory rootlets, which are purely sensory, combining to form the sensory root. The motor rootlets can be seen here, forming the motor root. The roots combine to form the spinal nerve, which is a mixed structure now. The nerve then splits up into a larger anterior ramus and a smaller posterior ramus. Now let's just discuss the relationship of the spinal nerves to the vertebral bodies and the intervertebral foramina. So let's take the help of this cartoonic illustration, which basically is a mid-sagittal section taken through this cranial cavity over here and through the vertebral column. You can see the bodies of the vertebrae in the front, so this would be the anterior side. The spinous processes can be seen at the back, so this is the posterior side. You can see the brain stem continuing down as the spinal cord. Right, so we know by now already that there are 31 spinal segments, which means that there are 31 pairs of spinal nerves, each spinal nerve popping off from its respective spinal cord segment. Each pair of spinal nerve, after it originates from the spinal cord segment, it's gonna then exit through the intervertebral foramen and innervate the peripheral structure. Now, an important concept which you have to understand over here is that there are each cervical spinal nerve emerges through the intervertebral foramen above its respective vertebrae. For example, if you look over here, you can see the first cervical spinal nerve exiting through the intervertebral foramen above the C1 vertebrae, and similarly the second cervical spinal nerve is going to pop off from the spinal cord, pass through the intervertebral foramen and exit it above the second cervical vertebrae. However, there is a discrepancy between the number of the cervical spinal nerves and the cervical vertebrae. There are 8 cervical nerves, but there are only 7 cervical vertebrae. Hence, the 8th cervical nerve, which is shown over here, that will emerge underneath the 7th cervical vertebrae, which implies that from the first thoracic spinal nerve and onwards, all the spinal nerves are going to emerge through the intervertebral foramen underneath their respective vertebrae. For instance, the T1 nerve is now seen emerging from underneath the T1 vertebrae. The T2 then emerges from underneath the second thoracic vertebrae, and so on. Right, so now from a functional perspective, the nervous system can be divided into a somatic and an autonomic nervous system. Students usually have a hard time understanding the anatomy of the autonomic nervous system, especially how it interconnects with the rami of the spinal nerves. Hence, we will talk in detail about the autonomic nervous system now. We will discuss the layout of the ANS in general, and we'll also see how the autonomic nervous system contributes to the innervation of the respiratory, cardiovascular, and the GI systems. Since the visceral, sensory, and motor neuronal fibers, they enter and leave the CNS in the spinal nerve along with their somatic counterparts. Therefore, let's have a quick recap of the anatomy of a spinal nerve. By now, we know that each spinal nerve originates from its respective spinal cord segment. You can see the spinal cord, cut section of a spinal cord inside the vertebral canal over here. This is the posterior spinous process of the vertebrae. The body of the vertebrae is in the front, so that's the posterior side. This is the anterior side. Right, so each spinal nerve originates from its respective spinal cord segment and is formed by the union of the anterior motor root with the sensory root at the back over here. The two roots, the motor and the sensory roots, they join together to form the spinal nerve, roughly at the level where the intervertebral foramen is present, and the spinal nerve then splits up into a larger anterior ramus and a smaller posterior ramus. Now, as far as the sensory information processing is concerned in context of the autonomic innervation, the visceral sensory neurons are the ones which bring in the visceral sensory information into the CNS, such as the spinal cord. These visceral sensory neurons, they travel along with their somatic counterparts as they run through the anterior or through the posterior rami, reaching into the spinal nerve and then running through the posterior sensory root over here to enter into the posterior sensory gray horn of the spinal cord. The autonomic motor outflow then can be categorized into two types: the sympathetic motor outflow and the parasympathetic motor outflow. Unlike the somatic motor outflow, which consists of only a single type of motor neuron exiting the spinal cord through the anterior root over here, the autonomic motor outflow comprises of two types of neurons: a preganglionic motor neuron and a postganglionic motor neuron. Let's take up the sympathetic parasympathetic nervous systems one by one. So starting with the sympathetic nervous system, the preganglionic sympathetic neurons, they have their cell bodies situated in the lateral gray horns of the spinal cord. This is a cartoonic illustration. So the lateral gray horns, they exist only in the spinal cord segments from T1 to L2, and this is actually shown over here in this cartoonic illustration. See, that's the brain, and you can see the brainstem continuing down as the spinal cord. These different color coded regions of the spinal cord represent the cervical, and then the thoracic, the red thoracic region, and then the lumbar and the sacral regions over here. The lateral horns exist only in the T1 to L2 segments of the spinal cord, which means they're not going to be present inside the cervical region of the spinal cord. They're not going to be present in the lower lumbar and the sacral regions of the spinal cord. Now, the preganglionic motor neurons, preganglionic sympathetic motor neurons, following their origin from the lateral gray horns of the spinal cord in the T1 to L2 segments, these preganglionic neurons, they're going to travel through the anterior root over here. Okay, they're going to reach the spinal nerve, and from the spinal nerve they're going to go into the anterior ramus, and through the anterior ramus they're going to these preganglionic neurons, they're going to jump into the sympathetic trunk. There's one sympathetic trunk on either side of the midline outside the vertebral column, situated literally on the surface of the vertebral column. Each sympathetic trunk contains a series of ganglia. You can see that cartoonic illustration of sympathetic trunk over here. Each sympathetic trunk outside the vertebral column contains a series of ganglia and is connected with the anterior rami of the spinal nerves via channels called the white rami communicants and the gray rami communicant. This can be much better understood by looking at this cartoonic illustration over here. So we can see the cut section of the spinal cord, one of the segments of the spinal cord, and we can see the preganglionic sympathetic motor neuron coming out from the lateral horn, passing through the anterior roots of the spinal nerve in that spinal cord segment, and then reaching onto the spinal nerve, and then the spinal nerve, and then running in the anterior ramus of the spinal nerve. These preganglionic sympathetic neurons, they're going to be then traveling through this structure here called the white ramus communicant, which connects the anterior ramus with the sympathetic trunk over here. So through the white ramus communicant, the preganglionic sympathetic neuron is going to reach into the ganglion of the sympathetic trunk at that respective spinal cord segment level. Now, from here onwards, this preganglionic neuron could take on following three possible routes. The preganglionic neuron could actually synapse with the cell bodies of the postganglionic neuron inside the ganglion of the sympathetic trunk, situated at the same level as that of its spinal cord segment of origin over here. After synapsing inside the ganglia of the sympathetic trunk, the postganglionic neuron then travels through a structure called the gray ramus communicant to re-enter into the anterior ramus at the same spinal cord segment level. So therefore, the anterior ramus is connecting with the ganglion through a white ramus communicant, through which the preganglionic neuron was traveling, and also through a gray ramus communicant, through which the postganglionic sympathetic neuron is traveling and entering back into the anterior ramus. From here onwards, these postganglionic sympathetic neurons can travel through either the anterior ramus or through the posterior ramus to provide autonomic innervation to the intended target organ. Right, so now what's the second possible route which the preganglionic neuron could take up? We know that the preganglionic sympathetic neuron that originates from the lateral gray horn of the spinal cord. We know that by now. And these lateral horns, we also know that they exist only in the T1 to L2 segments of the spinal cord. But what about the body structures which exist above the T1 level, such as the head and neck region, or those which exist below the L2 level, such as the pelvic structures? To reach out to these structures, the preganglionic neurons need to somehow reach into the cervical spinal nerves or into the lower lumbar and sacral spinal nerves. In order to accomplish this, the preganglionic neurons, once they reach the sympathetic trunk, right, following the same route, once they reach the sympathetic trunk through the white ramus communicant, they do not synapse over here with the ganglion at the same spinal cord segment level. So what I mean by that is that the preganglionic sympathetic neurons will travel through the anterior route to reach onto the spinal nerve and then into the anterior ramus, and from there through a white ramus communicant they're going to go into the ganglion of the sympathetic trunk. However, they will not synapse over here, rather they will either travel up or they will travel down in the sympathetic trunk. Usually the preganglionic fibers above the T5 spinal cord segment, so somewhere over here, somewhere in the midline, they usually, when they reach the sympathetic trunk, they can travel up over here, and the preganglionic fibers which are coming from the T5 segments and below the T5 segments, they once reach the spinal cord, once they reach the sympathetic trunk, they can descend down into the sympathetic trunk. For example, the preganglionic autonomic neurons innervating the head and neck region and the brain, they could originate from the T1 spinal cord segment. Upon reaching the sympathetic trunk, these then ascend within the sympathetic trunk to reach the superior cervical ganglion. These then synapse over here, and the postganglionic neurons, these postganglionic sympathetic neurons arising from the superior cervical ganglion, they then re-enter the spinal nerve via the gray ramus communicant. However, this time this is going to be a cervical spinal nerve. So the same story is going to repeat at a higher level as well. Just like we saw over here, the postganglionic neuron is going to pass through the gray ramus communicant to enter into the anterior ramus. The same thing is happening here as well, but it's just happening at a higher level. So once again, you can see let's say this is a higher superior cervical ganglion. The postganglionic neuron is going to pass through the gray ramus communicant and then enter into the cervical spinal nerve. Similarly, let's take another example where the sympathetic neurons, they have to innervate the pelvic structure, such as some of the viscera present inside the pelvic cavity, the bladder, for example, or the reproductive organs, for example. The preganglionic neurons, they could originate from the lumbar, let's say L1 or the L2 spinal cord segment. These preganglionic neurons, they, upon reaching the sympathetic trunk, they could descend inside the sympathetic trunk to reach to the sacral part of the sympathetic trunk. These neurons, which are still preganglionic, they could then synapse with the ganglion in the sacral part of the sympathetic trunk. The postganglionic neurons originating from the ganglion then could re-enter the spinal nerve via the gray ramus communicant. However, this time these would be the second to fourth sacral spinal nerves, right? So we can see that much more clearly here as well. See again, the preganglionic neurons, they're not going to synapse at the ganglion higher up over here. Rather, they're going to descend down, and then once they reach a ganglion which is much below the level of the L2 spinal segment, there the postganglionic neurons are going to go back into the anterior ramus of a lower lumbar or sacral spinal nerve through a gray ramus communicant. And this also tells us one more thing over here is if you look over here, see the white ramus and the gray ramus communicants, they're basically present at the level are present all along the length of the T1 to L2 spinal nerves. But the cervical spinal nerves, or the nerves above the T1 level, and the lower lumbar and the sacral spinal nerves, they only have a white ramus communicant, which allows for the exit of the postganglionic neuron into the anterior rami. The gray rami communicant, they're not going to be present over here. So the white ramus communicants are present along the entire, they basically are present along all the spinal nerves, but the gray rami communicants exist only for the spinal nerves from T1 to L2 level. Now, there's a third possibility as well, where the preganglionic sympathetic neurons, upon reaching the sympathetic trunk at their respective spinal cord segment level, they do not synapse in the ganglia. So they're just not going to synapse over here in the ganglia. Rather, they're going to travel straight through some branches of the sympathetic trunk, which are called as the splanchnic nerves. Right. This is a cartoonic illustration which shows you the splanchnic nerves over here. You can see these are the branches of the sympathetic trunk. The sympathetic trunk can be seen over here on the surface of the vertebral column, and splanchnic nerves are more or less the ventral branches of the sympathetic trunk. There are several types of splanchnic nerves originating all along the length of the sympathetic trunk. These are the greater, lesser, least, lumbar, and then the sacral splanchnic nerves as we move from above down below. So there are different types of splanchnic nerves at different levels. The preganglionic sympathetic neurons, they just travel straight through the splanchnic nerves to innervate various visceras such as the GI systems or the pelvic visceras and so on. You should also know that there are various autonomic plexuses situated close to the midline of the body, such as the prevertebral plexus, which exists along the abdominal aorta, for instance. And the reason why the reason why I mentioned the plexuses over here is that there are different ganglia situated within the autonomic prevertebral plexuses, and the preganglionic sympathetic neurons, which were traveling inside the splanchnic nerves, they synapse with the ganglia in the plexuses close to the abdominal aorta over here. And this is the area where the postganglionic neurons are then given off, and these then run to innervate the different target visceras. Some of the important ganglions are the celiac ganglion and the aorticorenal ganglion and so on. Now let's talk a little bit about the parasympathetic autonomic motor outflow. The parasympathetic outflow is craniosacral, which means that some of the preganglionic parasympathetic neurons originate from some nuclei in the brainstem and then exit the CNS via 4 different types of cranial nerves. These are the 3rd, the 7th, the 9th, and the 10th cranial nerves. Other originate from the S2 to S4 spinal cord segments and then travel through the pelvic splanchnic nerves to reach on to the pelvic visceras. Just to make a note over here that just don't confuse the pelvic splanchnic nerves with the lumbar or the sacral splanchnic nerves. The lumbar sacral splanchnic nerves are in the same region as well, but they are containing sympathetic neurons, whereas the pelvic splanchnic nerves, they contain parasympathetic neurons. Now, just like the sympathetic neurons, the parasympathetic neurons are also categorized into pre- and postganglionic types. The preganglionic neurons traveling, so let's start with the cranial outflow first, and then we'll jump over to the sacral outflow after that. Now, the preganglionic parasympathetic neurons traveling through the third cranial nerve, which is the oculomotor nerve. They are going to synapse with a ganglion called the ciliary ganglion, situated close to the orbit. The postganglionic parasympathetic neurons then will run and eventually innervate the iris and the ciliary muscles to help in mediating the light reflex and accommodation for near vision. We'll discuss about that in much more detail in the second year though. The preganglionic parasympathetic neurons traveling through the seventh cranial nerve, which is the facial nerve, these innervate the lacrimal gland and in the eye, and the submandibular and sublingual salivary glands in the oral cavity. Which are kind of shown over here, the submandibular sublingual glands here, and then the lacrimal gland close to the eye over here. The preganglionic neurons, they synapse with the pterygopalatine ganglion and the otic ganglion, from where the postganglionic neurons arise and then innervates these glands, the lacrimal gland and submandibular and sublingual salivary glands. The preganglionic neurons traveling through the ninth cranial nerve, they are gonna run through they're gonna run in the glossopharyngeal nerve, which is the ninth cranial nerve, and they will then synapse over here in a ganglion called the otic ganglion, and the postganglionic neurons arising from here, they're gonna then innervate a salivary gland called the parotid gland. So they're concerned with the salivary production as well. Lastly, the preganglionic neurons traveling through the 10th cranial nerve, which is the vagus nerve, they innervate the heart, the lungs, the tracheobronchial tree, the foregut, and the midgut up till the transverse colon. These preganglionic neurons, they travel through the vagus nerve to reach on to the autonomic plexuses, such as the cardiac plexus, the pulmonary plexuses, and the prevertebral plexuses located on the aorta. The postganglionic parasympathetic neurons then arise from over there, which are really short in length. They are situated close to the visceras, such as the GIT or the other pelvic or the other visceras, and then innervate them through those short postganglionic branches. Lastly, the parasympathetic outflow for the pelvic visceras originates from the sacral segments of the spinal cord. The preganglionic parasympathetic neurons, they travel through the pelvic splanchnic nerves, which are branches of the sacral plexus, which carry these preganglionic neurons to the hypogastric plexuses. The postganglionic neurons then arise from there, which are short and are situated very close to the visceras, which they innervate.